Darwin turned a few visual observations over a few years into a hypothesis on how all the diversity in living things had come to be. But he recognized a big risk to his ideas, and here's what he said. If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous, successive, slight modifications, my theory would absolutely break down. With that idea in mind, let's now take a look at the concept of irreducible complexity. The mousetrap is a very simple illustration of a machine that is irreducibly complex. It only has 6 parts, but if you take away any one part, you no longer have a working mousetrap. In fact, it wouldn't serve any purpose at all. All the parts have to be in the same place at the same time and properly aligned in order to have a functioning mousetrap. Well, now that you understand the idea, let's look at a highly complex molecular machine that is also irreducibly complex, the bacterial flagellum.
In Darwin's Black Box in 1996, Behe spotlighted and made famous a number of really interesting discoveries that had been occurring in biochemistry and cell biology over the last 2 or 3 decades. And what biologists, molecular biologists, cell biologists, microbiologists have been discovering is that at the level of individual cells, there are little tiny examples of nanotechnology, little tiny machines at work. The flagellar motor is the one that Behe made most famous. It's a rotary engine that powers a whip-like tail, a protein tail that functions like a propeller, and it moves the bacterium through liquid, enabling the bacterium to essentially track down its food supply. And this little machine includes a rotor, a stator, a drive shaft, a U-joint, bushings, bearings, and a whip-like tail that functions like a propeller. And the machine in some bacterial systems turns at 100,000 RPMs in one direction and can reverse direction on a quarter of a turn and turn 100,000 RPM in the other direction.
Bacterial flagellum is a true nanomachine, about 40 nanometers in size. It's amazing. I mean, E. coli, salmonella, which are kind of our model systems for the bacterial flagellum, can propel a cell about 20 lengths per second through a very viscous medium, like water, to these organisms. And you extrapolate that to human scale. 20 body lengths per second, 6-foot person, times 20, 120 feet per second. Mark Spitz or Phelps would be setting records with this type of propulsion.
It's hardwired into a signal transduction circuit that allows the bacterium to sense changes in the sugar gradient in the surrounding liquid.
This signal transduction system is actually a short-term memory system where the cell is, if it's going in the direction of an attractant, a nutrient that it can use to metabolize, it follows that chemical gradient. If it's a repellent, it will sense that and move in the opposite direction. So it's more than just this engine.
It's an extraordinary piece of nanotechnology. It's high-tech in low life. And so just by spotlighting these extraordinary pieces of nanotechnology inside cells, and the flagellar motor wasn't the only one by any means, Behe, in a sense, opened up a window for people. He opened up the black box of the inner workings of the cell and said, look, this is much more complex than anything that the early evolutionary biologists had envisioned. Darwin knew nothing of this type of nanotechnology in cells, and at the very least we've got to come up with an explanation for this.
Like the mousetrap, all the parts have to be in place at the same time in order for the machine to work. What happens if you take one part away? Well, it's not that you have lower performance, you have no performance. The machine stops working. It's completely useless. The idea that small changes over time accumulate to better and better performance falls apart completely when you consider the flagellum. Now, we could go on longer and look at other examples, but for the sake of time, we're going to change subjects again. We'll look at another cornerstone of Darwinian theology, mutations. As Darwin studied birds and animals, he observed that there were occasionally small changes from one generation to the next, and another change perhaps in the next generation. Now, he didn't know why, he didn't know about DNA, but his observations of those small changes formed the basis of his hypothesis. Today we know that the changes we see are the result of mutations that occasionally happen in the reproductive process from one generation to the next. While Darwin thought that accumulated mutations would lead to bigger and better, we know today that is not the case. The estimates about beneficial versus detrimental mutations vary widely, but here they are. On the low end, the estimate is one good mutation for every 10,000 bad ones. And on the high end, the estimate is one good one for every 10 million bad ones. It's pretty hard to imagine a bacteria becoming a bear working with those odds. Well, let's look at one example of what usually happens with a mutation. Our example is hemoglobin. Hemoglobin is the red blood cell. It's the cell that picks up oxygen in the lungs and carries it throughout the body. The picture is a normal red blood cell. The mutation we're talking about is called sickle cell disease. This is a rendering of the hemoglobin protein. It's composed of 4 amino acid chains, 2 alpha and 2 beta. The 2 alpha chains each contain 141 amino acids each. They're normal in people with sickle cell disease. The beta chains each have 146 amino acids, but the 6th amino acid in the beta chain is different in people with sickle cell disease. The normal chain has glutamic acid in the 6th position. In the mutated chain, the 6th position has valine. That's the mutation. And that's the only difference between a normal protein and a mutated protein. Out of a total of 474 amino acids that make up this protein, only one error occurs. And what is the result? Sickle cell hemoglobin. A deformed hemoglobin molecule, and the result is clogged arteries, which can lead to blood clots, stroke, and death. Now, fortunately, mutations are fairly rare, but research and record keeping have shown that almost all of them are detrimental to the host, not helpful. And although it's hard for people to give up on an idea on which they've built their careers, some scientists and academics are beginning to walk away from Darwinian theology. This is Dr. David Galertner. He's a professor of computer science at Yale University, and he says, "There's no reason to doubt that Darwin successfully explained the small adjustments by which an organism adapts to local circumstances, changes to fur density, or wing style, or beak shape. Yet there are many reasons to doubt whether he can answer the hard questions and explain the big picture, not the fine-tuning of existing species, but the emergence of new ones. The origin of species is exactly what Darwin cannot explain." Well, once again, I feel like we're seeing that the whole materialist theology is built on sand. Well, in my next letter, we'll look at paleontology, the study of fossils. In the meantime, sweetheart, continue to work hard at school, and remember, we love you. I'll talk to you soon.